Hi, everybody. So, online people, we just watched the link that's there in your notes. Um, I think that it's the same one that we saw, I think, the first day of class. You people have learned so much. Like, at least some of the things where you're now like, oh, I know what that thing is. Yeah, go us. Also, with the you've learned so much, I was going through to make today's PowerPoint of doom that has like all the things. We've covered a lot of ground, so you should congratulate yourselves that you're still showing up either virtually or in person, and that like there is a ton you know. So yeah. Things to remember, because all I do is remind people about things now. I don't know. I become annoying even to myself. You have your lab exam if you've already taken it, great. Today is the last time to take your lab exam. Yes? Nods? Okay. Taking your lab exam. Other things while I'm thinking about it. Lab exam, the class paper is due today with a caveat. You know how like if you're driving on the highway and I use this with somebody else to kind of explain this idea. Driving on the highway, it says 70. If you're going 75, nobody cares. If you go 85, the cops will get you for being 15 over the speed limit. Yeah? Paper is due today. Get it in by Tuesday at midnight, and I don't care. But because it's due today, if you try to get it Tuesday at midnight and your computer blows up and a hedgehog eats your paper and all the things, I will be unsympathetic and crabby. Okay? I'll be like, sounds like a personal problem. Okay? Yeah? It is hard for me to be crabby to you guys because I like you, but like I have to draw the line because otherwise I never get this done. VIA is how you have to turn these things in. It's because this is a gen ed class. I'm supposed to teach you something about science. So your paper where you learn something about science has to show up in VIA. And people besides me, if they ever want to, are going to read these. Sometimes they actually do. So you're turning them in on VIA. The way that happens is if you go to Owlnet. Oh, good grief. Sorry, I'm signing it over here. You go to Owlnet, online people, you also go to Owlnet. You go into your coursework. You should see bio issue paper. You click on the link. You're here. You click on the link. It takes you to VIA. If you don't already have a sign up, you make a sign up. And then you just upload it like you would attach a file to an email. You can also, if you really want to, you can type it out there. But that makes me feel scared, just in case, like, just have it someplace else too, okay? Like, if you want to copy and paste, fine, but like, save it someplace that you have control. If you, and this, officially, I frown on this as an idea, but as a human, I understand. If you're still maybe working on your paper and you're not going to get it done today, get it done. But I'd suggest you at least go and see if you can get onto VIA, just so then you can put this off until Tuesday and just feel more calm about it.
If we click on the link you send, that works too, right?
Yes. Okay. Yeah, the link gets you here.
Okay.
Yes, the link gets you here, but just make sure you can sign up, because the past 2, maybe 3 semesters, I've always had somebody 15 minutes after to do say, I can't figure out how to get onto the VIA. Okay, so just, most people have no problem, but there's always somebody, and that's fine. If you're that somebody, that's cool. But be that somebody early, not late. Yeah. And with this, I would like to give you, for this and for your lab paper, I want to give you tons of points for it. Exams, I have no problem being like, haha, it should be hard. For these ones, for the papers, just don't give me a reason to take points off. Have peer-reviewed sources, don't plagiarize, have it make some level of sense, but don't spend hours crafting beautiful sentences, okay? Just get it done. Yeah? Okay, like I'm serious on this one. Peer-reviewed sources make some level of sense, not copied. Great. Same goes for the lab thing. Okay. I'll be sending you like all the emails over these next few days just to make sure we're all on the same page of stuff. But we're doing okay. You have the exam on Monday, so you're not coming into class on Monday. If you'd like to take the exam early, you're welcome to. It's open now. It closes Monday at 11:55 p.m. So just take it sometime. So far, so good. I have so many slides to go over with you, but we have time. Do you have specific questions we should jump to? Or the way it's currently organized is the last third of the class is in slides. And then, so like, picking up from where we started after exam 2, the last third, and then it starts at the very beginning of the semester through the first 2/3. Does that work for us? I'm gonna talk fast. Stop me or slow me down if you want to. Again, review all your notes, all the things, but just these are the greatest hits. And I was impressed with like, they've covered so much. Nicely done. The cell has what's called the cell cycle. Here in G1, the cell's just like chilling and doing its normal cell thing. If it hits the G1 checkpoint and moves on, it's going to divide. When it gets to the S phase, it's going to synthesize DNA. When it gets to the M phase, if it passes the G2 checkpoint and gets to the M phase, it's going to actually go into this mitotic phase where it's going to pull apart. And then you have 2 cells. Yay. Meiosis and mitosis are different. Which one's happening like all the time in pretty much all the cells? Mitosis, yeah. Mitosis is happening all the time. It's happening in mitose. Yeah, it's happening all the time, right? It's not trying to have babies. It's just trying to get more cells, right? These daughter cells are going to look genetically identical to the parent cell, barring mutations. They're going to look identical to the parent cell. Meiosis is going to be the special kind that you only sometimes do, right? Only germline cells who try to form gametes. These daughter cells are going to have half the chromosomes of the parent cell. They're going to be haploid. And they're going to be genetically unique in that crossing over got to have happened. So these daughter cells are all different from each other. They're different from the parent, and they're different from each other. Great. Okay, well, with the cell cycle, on to Mendelian inheritance. Whew. So many, I realized we spent like weeks talking about these things, and now I'm like, one slide. Complete dominance, right? Does a dominant allele have to be more common in the population? Right, it does not have to be more common, right? The allele for dwarfism is dominant, right? But it's still relatively rare, yeah? Does the dominant allele have to be better, more normal? Also no, right? Dominant just means if you have 1 copy of it or 2 copies of it, it looks the same, right? Whereas recessive, you only are going to see if you have 2 recessive copies. Cool. Incomplete dominance is the idea with something like here with these little snapdragons, where you have the dominant form, the red, you have the recessive white, but the heterozygote looks halfway between either the dominant or recessive homozygote. Okay with that? But there's not a gene that says make pink. There's a gene that says make red or don't make red, but it's just incomplete dominance because if you only have one copy, it doesn't completely cover. Yeah? Codominance is when you see both phenotypes expressed in the heterozygote. So if you're homozygous for A-type blood, you have just A. If you're homozygous for B, you have just B. If you're heterozygous, you have A and B shown. Multiple alleles is you've got several different versions, right? For blood, you could have A, B, or O, right? For eyes, you can have blue or green or laser vision or whatever it is you have, right? Brown, who knows? Pleiotropy is when one gene has lots of effects. So instead of it just makes one pigment, it's like, well, it does all the things. Epistasis is when one gene is going to determine how another gene behaves, right? So it's going to be these complex interactions. Polygenetic inheritance is it takes more than one gene to make the phenotype, right? So lots of things going into, say, something like height, right? This is not just one gene, a tall gene and a short gene. You have lots and lots and lots of genes for that. I want to show you something on the board really quickly. I think we're doing okay on time. Speaking as somebody who finished your exam 3 last night, I had been working on it. I've never fallen with this, but it's been close. Oh, that's not high. Okay, if you have some, online people, can you see this? No, fine. If you have some convoluted question like this, where it's like, okay, you've got parent, is green easier to see than white? Which is easier to see? No difference? Okay. If you have some parent that's like this, and that parent mates with someone who looks like this, I don't know, something like that. And the question asks, what's the likelihood that the offspring has this? Okay, you've got something like this. Okay, so you're trying to say, what's the likelihood of this given these parents? Do not. Please don't, like, draw one huge 64 grid Punnett square and try to put, no. Not enough time. Is there a fast way to check this? Should we do it really quickly? We can do each of these individually, remember? If we had this parent and this parent right here with just the A's, what's the likelihood of this? You can draw one little tiny dinky one if it helps. Okay, what's the likelihood of big A, little A? Yeah. 1/2 there. If you have these heterozygotes, big B, little b, big B, little b, we could draw it out, but what's the likelihood of getting 2 big B's?
24.
Yeah, it's 1/4. Getting 2 little C's here?
75.
This one's a little tricky. We'll look at it here. This one could be either little c or little c, big C, little c. They all look the same. Oh no. Let me try that again.
Sorry,
It's 50. Right, because this one can give either, and this one can only give little c, so it's half. Okay, so then you look at this and you're like, okay, 2 times 4 is 8, 8 times 2 is 16, 1 times 1 is 1, 1 times 1 is 1, so 1 out of 16. Do you see that? Don't try to do like a huge grid, just each one individually. That cool? Find this? Okay. Next thing, that. Chromosomes are what's holding this genetic information, right? Chromosomes, just because they came together from a parent, they don't remember what their buddy chromosomes were. They can sort independently, right? And they split up. Fair enough? Cool. Don't freeze, computer. There we go. This thing I want to throw on the board again. Come on. Remember the cat situation? Yeah. Sorry. I feel like the phrase "remember the cat situation" can mean many, many different things, given the time. Most of the cells in whatever we're going to be in mammals are expecting 2 of every single chromosome. The exception are the sex chromosomes, right? You're only expecting 1 functional X chromosome in each cell. If you're male, you only have 1 functional X chromosome, so cool, you're set. If you're female, 1 X chromosome is going to collapse down. So if there's a gene on the X chromosome, you can end up with interesting things like this. Yeah? This one I want to do on the board as well. Because my goal is to be as covered in chalk as humanly possible. Let's say you've got a tortoiseshell cat. Do you know if tortoiseshell has to be male or female? Mm-hmm. Yeah, it has to be female, right? Because you've got this thing for coat color is on the X. So if you have X with orange and you have an X with black, this cat's tortoiseshell. If that cat mates with a black male cat, what kind of offspring could they have? So black male cat, how many X chromosomes? 1. So you've got X is black. The other chromosome is going to be the Y chromosome. It does not have this gene there, right? So this cat right here, this could be an offspring. Male or female? Female, what color? Yeah, we've got a female tortoiseshell cat, right? This one right here is also female, but she's black, right? This one here is an orange male, and this one here is a black male. Do you see that? So a black male cat and an orange and a tortoiseshell female cat can have kids that are black, orange, tortoiseshell, but they can only have, like, any kid that's completely black is, okay, they can have black sons or daughters. Orange kittens have to be male in this scenario, and tortoiseshell kittens have to be female. Do you see that? Questions or concerns? Yeah? We're okay, okay. There is a question very, very similar to that on the exam. All right. Next, the genes are physically located on the chromosomes, right? So genes that are on different chromosomes completely assort independent of each other, right? But if 2 genes are on the same chromosome, they come together, usually. They don't have to, though. If you've got 1 gene right here on the chromosome and another gene right next to it, they show up together, typically. If you have a gene on one place in the chromosome and way far away, you have another gene. They come together, but not always. Why don't they always come together? Are we okay that the crossing over is going to mix stuff up? And the farther apart you are, the more places you could cross over and have it mix up. Whereas if you're close together, you have to cross over right there to mix up. Yeah? Am I going too fast? Are we okay? We have a whole semester to cover in 50 minutes. Again, I'm not trying to teach you all these things, it's just kind of jogging the memory. Sometimes chromosomes don't split up appropriately. It's called non-disjunction. You can end up with either having an extra or missing a chromosome. This is a problem for pretty much everything. The X chromosome is the one that's the least concerning, because the X chromosomes can collapse down and just not do much. So if you have 3 X's, it's not really a problem. We're okay with this? Also, if your chromosomes get broken, like they're trying to do crossing over but they just don't come back together, that can be a problem too. Right? If there's just chunks missing, that's not good. Okay, deep breath. What's this? Besides cool? It's DNA, yeah! It's our little DNA molecule, buddy. The sugar phosphate backbone, the nucleotide, right? Different nucleotides. You can have whatever nucleotide on whichever strand, but the other strand needs to have a complementary one, right? This strand, like one arm of DNA, one strand of it, this is all covalent bonds, right? Covalent bonds the whole way down. Its molecule, it's holding on tight. One strand versus the other strand, those two, they're being held together by hydrogen bonds, right? So it's easy enough to pull the two strands apart. It's hard to pull the strand, like, to snap the strand. You've got the 5' end and the 3' end there on that strand. The other strand is running opposite to it. It's running counter to it. It's flipped. This whole thing coils. So far, so good. We're feeling comfortable with this. These are, you can have individual nucleic acids come together to make nucleic acids. Do you remember this experiment? It's beautiful. You grow up DNA in something heavy, so your DNA is more heavy than normal. And then you pop it into just like regular solution. You let it replicate once, and you see that the whole system is semi-conserved, meaning one arm stays exactly what the original was, and you build a whole new arm. So then if you do it again, you end up here because you, again, pulled apart, you keep one of the originals that you pulled apart and add in new ones each. Okay. People seem underwhelmed by this experiment, which is fine. Just like, yep, that's nice. Whew. What's happening here? All the things, right? What's helicase doing? Unzipping, same thing, yeah, it's pulling it open. What's topoisomerase doing? Yeah, making sure it doesn't get coiled too tightly, right? Because like, right? It's gonna relieve the pressure there. And that's topoisomerase. The primase is adding a primer. The single-stranded binding proteins are keeping the whole thing from like sticking back to itself. The replication fork is where that thing's actually opening up, right? As you're adding stuff to this, you have to add to the free 3' end. There's no other place this little nucleotide can stick. It needs that OH group to be able to stick to. The 5' end, it does not have it. It can't do that. So it can add right there. DNA polymerase is what's doing this. Right? DNA polymerase is, adding these on. Free 3' end. For the final, it's it's going to be more big concept stuff, right? This exam is going to have a lot of like 5' or 3' that kind of thing. Final is going to be things like, what makes DNA? DNA polymerase. What makes RNA? RNA polymerase, right? Yeah. Whew. A lot happening here. Briefly, what's the difference between the leading and the lagging strand? Here. DNA is being replicated, by the way. Yeah, the lagging strand is made in chunks, right? So the leading strand is getting built in the same direction that the replication fork's opening, right? So just keeps going. The lagging strand is made in these chunks. They're both happening equally fast. They're both equally good. It's just lagging strand is in chunks, leading strand is one continuous in the direction of the replication fork. Oh, we've got ligase here, adding things back together. What's not shown on here is, is nuclease. Nuclease is what's going to proofread this and cut out bad bits. So if something's wrong, it's like, whoa, 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 whoa. Before we finish, we need to cut this out and fix it. So that's nuclease, it's cutting it out. Remember this thing about, like, being mortal? I know, sorry. The ends of linear chromosomes get shortened every time you replicate. So how do you solve this problem? Aside from accepting the inevitable hand of fate. Yeah, so on the ends, you have sacrificial nucleotides, right? They're called telomeres. And if those get shortened up, they don't mean anything besides they're just there to protect the edges. Fair enough? This is the same thing, so less dramatic. If you've got the cousin who's cutting your hair, and you know she always cuts it too short, you say, I want it here. You actually want it to your chin, but you tell her you want it to your shoulders just because you know you've got some sacrificial stuff there. Alright, chromosomes, they may have protein and DNA. DNA is holding the genetic information. Proteins are keeping this from being a huge mess, but they're both important. Going from DNA to RNA, it's transcription. You've got RNA processing in there as well if you're in a eukaryotic cell. Going from mRNA to a protein, it's called translation. The ribosome's going to get that done. This slide you should know cold, right? You could put any of the molecule names and any of the, like, where they go. Beautiful. I feel like I'm beating this to death, but you've got DNA, you read that to go to that's transcription, you end up with mRNA. You read mRNA in codon chunks, 3 nucleotide chunks, which gets translated into amino acids as you build this protein. Let's say you see this codon UUU, it's for phenylalanine. What's the anticodon that transfer RNA needs to have to match this? What's the anticodon that matches that codon? AAA, are we fine with that? It's the complementary one to what's shown there. Beautiful. Okay, this slide is the only one that, like, has so many words. You've got this universal code. Okay, 3 big things about the code for DNA or RNA. It's universal, meaning any life form, I realize I sound like a space alien when I talk about this, but any life form is using the same code as all other life forms, right? So mold is using the same code that elephants are using, right? There's a lot, the code, what that code says is different things, but this code's the same, right? So a children's book and your textbook say different things, but they're using English regardless, right? So that code's universal. The code is also redundant, but not ambiguous. Which means, so if your name is Jonathan, right? Maybe your family calls you Jonathan. At school, they call you John, and your grandma calls you JJ, okay, for whatever reason, right? That means you've got redundant names. You have 3 different names that all mean you, right? So for this, for phenylalanine, UUU or UC both mean the same thing, phenylalanine. It's redundant. However, it's not ambiguous. Ambiguous would be if the same name, if many people have the same name, if your parents had named all 17 of their children Jonathan, that would be ambiguous. Because then you say Jonathan, you don't know who they're asking, right? Code is redundant, more than one way to say something, but not ambiguous. This code only codes for one thing. Everyone solved on that? Yeah. Questions? Nope, okay. Yep, and it's universal, woohoo! All right, when you're making this mRNA, you're going to use RNA polymerase. The DNA is not destroyed when this happens, it just is read, and you make RNA. Okay, I lied. There's another slide that has lots of words. You can do alternative RNA splicing. You add bits on the ends to protect them, that's great. But you can have one gene that can code for lots of different proteins because of how you arrange the parts. Parts that are expressed are called the whats? The exons, yeah. Parts that do not make it out of the nucleus and get completely cut out are called the whats? The introns, all right? They are going to stay inside the nucleus. They're never going to be expressed. Yeah? Great. I really like RNA splicing, which means there are now 2 questions on the exam about it because I think it's adorable. I don't know why I like it so much, but I just think it's cool. Here's that little transfer RNAs. They've got the anticodon that's gonna match, it's gonna fit chemically with the mRNA. There's amino acid binding spot. It's carrying an amino acid that matches with its anticodon. This stuff is these polypeptide chains are going to become protein, get built by the ribosomes. Stop codons tell the thing to stop doing its stuff and to disassemble. And then this is the whole process there, right? You've got transcription up in the nucleus. If you're in a prokaryotic cell, it's not be the nucleus because it doesn't have a nucleus. The exons get strung together, the introns get cut out. mRNA leaves. The ribosome reads mRNA, uses transfer RNAs to bring amino acids to this, and builds this growing polypeptide chain in a process called translation. Whew. Messing this up is a problem. If you have a substitution at the DNA, that substitution, that message gets carried through each next step. That can mean lots of different things. Okay, apparently I have lots of slides that have words. Anyways, if it codes for the same amino acid as before, not a problem. If it codes for a different amino acid, that's kind of a problem. If it codes for a stop codon, that's a big problem. And if you just add or delete nucleotides, that is a huge problem because you're off. Everything's going to be wrong. Questions? We're good? Deep breath. That was the last third of the class. In 15 minutes, we're going to go through the first 2/3 of the class, okay? Woohoo! But ask questions, right? This, you should feel very much like we're amazing. Levels of organization in biology, atoms are smaller than molecules, molecules are smaller than cells, right? By this point, seeing that like oxygen is represented as O probably doesn't surprise anybody. Old news. Bonds, do remember something about bonds? Right, you've got covalent bonds that are sharing electrons, polar covalent bonds where they're sharing electrons but some particular atoms are sharing more than others, and ionic bonds where they've got opposite charges and they're attracted. Hydrogen bonds where you have two separate molecules, but the hydrogen ion of one molecule feels the electronegativity of the other molecule, and they're held together. This isn't as strong as something like a covalent bond, but they do have an attraction, right? We're good. Water is all sorts of fancy. It sticks together. It sticks to other things. It gets bigger when it freezes. Come on. It gets bigger when it freezes. It's really good at holding temperature, right? It's really good at taking a long time to heat up or a long time to cool down. And it also is a pretty good solvent. Water is superpolar. It's got these molecules polar. It's got these polar covalent bonds. It can hydrogen bond with itself. It can hydrogen bond with other things. It can tear apart something that's polar really, really well. Yeah? Water's beautiful. Acids and bases exist. The more acidic something is, the lower the number on the pH scale, right? So 0 is very acidic. The higher the number, the more basic it is. 14 is very, very basic. 7 is neutral. In acidic solutions, you've got more free hydrogen ions. In basic solutions, you have fewer free hydrogen ions. You've got more OH ions. Everyone's good? Carbon's also all sorts, so water is all sorts of special. Carbon's all sorts of special. Carbon is really good at making friends. It has 4 valence electrons. It can make 4 bonds. It can bond with carbon. It can bond with hydrogen. It can bond with oxygen. It can bond with pretty much anything. And it can make some really, really, really large, complicated molecules. It's beautiful. There are functional groups that keep behaving certain ways. That's great. They show up. These large, complicated molecules that are based on carbon come in 4 main types. You have your carbohydrates, right? So like sugars strung together as a polymer. Yeah. You've got lipids, which aren't a polymer, but they'll stick together. You've got proteins, which are a bunch of amino acids strung together in a polymer. You've got nucleic acids, which are a bunch of nucleic acids strung together in a polymer. All of these are based on carbon. Lipids you can end up with, if you've got phospholipids, you end up with a phospholipid bilayer, something that looks like this. You've got the phospholipid head that loves water, the hydrophobic tails that don't love water. They'll form up into this bilayer. Yeah. Amino acids come in 20 different versions. The amino acid part is the same for all of them, but the side group, that's what gives them their own special, unique, this is the amino acid I am identity. These amino acids can stick together in any combination, right? You can have histidine, arginine. You can put them together in any order. When you string these together, you end up with a polypeptide. Once you string those together and fold them, you've got a protein. The organization happens in a series of steps. So you string amino acids together, that's primary structure. You have them coil into beta coils or pleat together in alpha coils or beta pleated sheets. Right, so you have that secondary level of organization. Once you fold it into like a 3D doing something, it's tertiary structure. And then if you put a couple of these tertiary structures together, you end up with quaternary structure. but tertiary structures where most proteins are like doing their thing. And proteins really, really, really need to be in the shape they're supposed to do, otherwise they don't work. You need this polypeptide to be folded into the enzyme or into the structural form that you need. If you unfold it, say by hitting it with a hammer or throwing acid on it or cooking it, you denature the protein. It's not in the shape that works anymore. All right. Prokaryotic cells don't have membrane-bound organelles. Eukaryotic cells do. All cells are going to have a plasma membrane. They'll make cytosol. They'll have chromosomes. They'll have ribosomes. That membrane can let stuff pass through it. It's semi-permeable. Things go down their concentration gradients. It doesn't take energy to do this. You just have to have the membrane permeable to the thing that goes down concentration gradient. Water can go down its concentration gradient. It's called osmosis. So if there's way more water on that side because there's fewer solutes, water flows towards the side that has more solutes because there's less water. Great. Enzymes are going to make a lot of reactions happen faster. Processes happen because they're spontaneous. So a spontaneous process is going to occur without adding energy into it. It just can happen. Doesn't mean this has to happen quickly. It just means you don't have to pay for it with energy. Those processes are going to have negative delta G's. You can use those to harness those to do work. So flowing from water flowing down, it's negative delta G. You can harness that to power your little light bulb. To get work done, to get other things done, you harness spontaneous processes. So you capture energy, say, from either the sun or from food, and you make ATP. And then ATP going from ATP to ADP in inorganic phosphate is a spontaneous reaction. You harness that reaction to get work off. Enzymes are not going to change whether or not this is spontaneous, but they will make it happen more quickly. So going from reactants to products has the same delta G, but the higher the transition state, the higher that lump is, the less likely this thing is to happen. So enzymes are going to help lower that activation energy, and it's going to make the reaction happen way, way faster. Yeah? Okay, whew. We're doing fine. In the circle of life kind of thing going on, you've got energy flowing into the environment, into the ecosystem, via light. Chloroplasts, so photosynthesis is going to capture this. You're going to build up complex organic molecules. You can release that energy by breaking up those complex molecules in, say, mitochondria. You end up with carbon dioxide. You can then use that carbon dioxide plus the energy that comes from sun to build back complex molecules. In everybody's all-time favorite week of the semester, we did this. Yeah? Yeah. Cells want to break down complex organic molecules. You've got glycolysis, you can make a little bit of ATP doing that, you break it down. Going through the citric acid cycle is going to break this down. You're going to get rid of carbon dioxide. It's going to carbon that was in you as some sort of sugar or something is going to leave as carbon dioxide. You get those high energy electrons. You use those high energy electrons to harness that energy to make ATP. You're using those high energy electrons to pump hydrogens across the membrane in mitochondria. They then fall back through ATP synthase, and that powers the formation of ATP. Deep breath, we're doing okay? Great. Kind of the reverse happens in photosynthesis, where instead of going from a complex molecule, breaking it up to get carbon dioxide off, but to capture energy, to release energy, you capture energy from light. Excuse me, my nose is itching. You capture energy from light, you take in carbon dioxide, you use that energy from light to put together these carbons to build up a complex organic molecule. So far so good? Okay, that's the entire semester in a nutshell. That does leave us with exactly, I think, 5 minutes. 3 minutes. Questions? In the last 3 minutes of class? These notes are posted in case that helps. Again, this is not all the things you need to know about all the things, but the idea is to kind of like jog your memory. I will send you emails, which sounds like a threat. That's supposed to be nice, but sounds like a threat. Yeah. Oh, it's a good class. I look forward to seeing all of you on either Monday, Tuesday, or Wednesday of finals week and learning all about your cool projects. It's going to be great. Yeah, see, have a great Friday. Try not to get the plague over the weekend, etc.